This high-speed flight through the interior of a living cell reveals a bustling, microscopic metropolis. To keep a city this active alive, it requires a constant, relentless supply of physical building materials. This creates a massive logistical problem. The cell has to move all of these essential materials across its vast watery cytoplasm without them getting lost. The endoplasmic reticulum, or ER, solves this. It is a sprawling, highly interconnected superhighway. The ER operates simultaneously as the cell's heavy manufacturing hub and its primary internal distribution network. Without this continuous system of channels to produce and ship goods, a cell cannot build its own borders, communicate internally, or defend itself. Looking closer at the physical architecture, the ER forms a continuous network of membrane-bound tubes and oblong bags, which biologists call vesicles. This side-by-side diagram compares a cross-section of the ER membrane on the left to the cell's outer plasma membrane on the right. The biological material making up the ER's walls is structurally identical to the plasma membrane guarding the outside of the cell. As this glide reveals a heavily textured exterior surface, we enter the first distinct territory, the rough endoplasmic reticulum. It earns that rough designation from the thousands of tiny particles attached directly to its surface, known as ribosomes. Ribosomes are the active manufacturing plants of the cell, and they are present in all active living cells. The job of these ribosomes is to manufacture proteins. Once a protein is constructed on the surface, it drops directly into the internal hollow channels of the ER for immediate shipping. The rough ER acts as a dedicated, high-speed supply chain, moving these manufactured proteins out to various regions of the cytoplasm or deep into the nucleus, depending on what the cell needs in that moment. Curious, as this environment transitions away from the rough exterior, we enter a sleek, purely tubular landscape. This is the second territory, the smooth endoplasmic reticulum, which lacks ribosomes entirely. Because the smooth ER lacks ribosomes, it has a different manufacturing purpose. It produces vital fat molecules called lipids. This schematic demonstrates membrane biogenesis, which is how a cell physically builds and expands its outer boundary. Proteins from the rough ER and lipids from the smooth ER travel through distinct channels, merge together, and actively fuse into the existing cell membrane to expand it. Membrane biogenesis occurs across almost all cells, but the smooth ER also develops specialized functions depending on the specific animal and cell type. In vertebrate animals like humans, the smooth ER inside liver cells acts as a massive filtration plant that detoxifies poisons and drugs that enter the body. By housing protein construction, lipid manufacturing, and chemical detoxification all under one continuous roof, the ER operates as a highly adaptable multi-tool organelle. This slow, vast pullback shot revealed the entire illuminated ER network. Despite having different appearances and functions in its rough and smooth territories, it always operates as one continuous, massive structure. The immense size of this network provides a cytoplasmic framework, a physical surface area required for life-saving biochemical reactions to occur inside the cell. Some of the proteins and lipids manufactured here deploy as active enzymes and hormones, circulating outward to regulate the body. By generating physical building blocks of the cell and delivering them exactly where they are needed, the ER functions as the primary constructor of the cell's tangible reality. It is the living framework of microscopic life. Before we leave this network, let's review two critical facts from the expedition. First, what is the exact physical structure of the ER? And second, what specific type of material is primarily transported through the ER channels?